Thank you
Excellent.
for everyone who's stuck around so far. I'm going to talk to you about some of the work I've done in the lab of Srinjan Basu, who's down at Imperial now. We're really thinking about transcription factors here, which are key regulators of sulfate decision in early development, particularly two important transcription factors for the establishment of pluripotency in the early mouse embryo, SOX2 and NANOG. So NANOG is really necessary for the establishment of this pre-implantation version of pluripotency where SOX2 is upregulated and then persists past, which is the post-implantation flavor of pluripotency. So we kind of have this interplay between different transcription factors when they're expressed, at what levels. So this got us thinking about the different types of strategies that transcription factors use to target chromatin. And that's at the level of concentration, the actual binding kinetics, and importantly, forming regions of high density. So things like NANOG and other transcription factors have these very large, low complexity domains sticking off of these well-conserved DNA binding domains. In particular, NANOG has this tryptophan repeat domain, which has been shown to interact with other NANOG molecules in a homo-oligomerization process. So the first thing that we addressed was what are the relative expression levels of NANOG, that question about concentration. And here we're using mouse ES cells with our favorite transcription factors of interest tagged with a halo tag, which allows us to label that stoichiometrically with a bright fluorescent dye. So here, what I hope you can see by eye is that NANOG is lower expressed than SOX2, and this is also true when we actually go through and do the quantitative immunofluorescence. So the first take home here is that SOX2 is more highly expressed than NANOG at the protein level. So the next idea that we wanted to address were the binding kinetics. So this is twofold. So the first question is, how long do transcription factors remain bound to chromatin? And the next one is, what fraction of transcription factors are in a chromatin-bound state? So if we take the first question, how long do transcription factors remain chromatin-bound? What we're able to do is single molecule localization microscopy, again using that halo tag, but now instead of using a constitutive fluorophore, we use a photoactivatable where we only activate a few at a time. And that's what you're seeing over here with these dots popping up on the screen. So we use something called double helix point spread function single molecule localization microscopy. So unlike conventional Palm or Storm, where you have a single dot that gives you the XY position, we split that single dot into two dots, and the angle between the two dots gives us the Z. So we're actually able to get 3D position within the cell. And what we're doing here is we're taking long exposure, which motion blurs out all of the moving proteins. So we're just retaining those that are stationary within that time frame. And then if we do a time projection of this video, what I hope you can see are individual nuclei and individual nice double dots. So to actually look at this, you can't just say, okay, the fluorophore is there, and then once it goes, it's gone. Because a dissociation event and a photobleaching event look identical within the data. So the way we overcome this is through time-lapse imaging, where we add in delays or gaps between each individual acquisition. And what this allows us to do is to not only just image for a short period of time, where if we do continuous imaging, we're only capturing a localization for, about the order of seconds, we can actually move up to the order of minutes. Taking these information together, we can start to build up distributions and a simple mathematical model of exponential decay, and put these together to actually make sense of how long these proteins sit in their location. So we take our simple effective decay and split that into two components, a bleaching rate and a dissociation rate, and solve a linear equation. And when you take each one of those exponential distributions, we're then able to convert that into a single data point and fit lines to that. And from this, we can calculate that the bleaching rate is approximately 4 events per second. And we can actually go and calculate the dissociation rate and take the inverse to give us the residence time, showing us that nanog binds chromatin approximately twice as long as SOX2. So moving to the next question, what fraction of transcription factors are in a chromatin-bound state? So for this, we have to move to short exposure imaging, so about 20 milliseconds. So instead of motion blurring out all of those images, what we want to do is we want to capture the moving ones and the stationary ones. And for this, we turn to a higher density imaging technique called light field. So instead of splitting the localization into 1, we split it into 7. And what you're seeing here are the 7 fields of view that the image has been split into through the micro lens array here. And then what you can do, you can do another time projection, and you can see the individual nucleus, and then happy localizations. So these are the same localization across the different fields of view. So what do we do with this information? We first extract the localizations. We then do some particle tracking to connect the dots. And finally do subtrajectory classification to see what parts of that trajectory are stationary or moving. And from that, we get trajectories that look like this, where you can see by eye that this looks like it's moving, which we've labeled freely diffusing, and this looks like it's stationary, which we call chromatin bound. And we can calculate parameters like diffusion coefficients and a parameter called the anomalous exponent, which tells us about the type of motion that these particles are undergoing. And importantly, what that allows us to do is to say what percentage of the observed molecules are in a chromatin bound state. And we see that both of these exhibit about the same level of being in that chromatin bound state. So the last question here is about the spatial component. So we've gone through and we've gotten all these nice classified trajectories out. Is there some type of higher order structural organization happening? When we go in and we take the centroid and subset these into just the freely diffusing or the chromatin bound trajectories and take the centroid of those, and then say, how close are you to each other, right? So we do something called a Delaunay triangulation, which is basically connecting the dots by triangles. And that gives us a density metric of a median edge length over that triangulation to say, if you have a small edge length, you are more closely packed than if you have a large edge length. And from this, we do some Gaussian mixture modeling to ask the data how many populations exist within there. In the instance of nanog, we see that there are 2 populations here. In the instance of SOX2, there is not. I draw this line here to show you that there is a shift to the shorter edge length, showing that the nanog molecules are more packed in. And when we label those by orange being densely packed, red by being less densely packed, we see that we get these little clusters. So what this tells us is that nanog is, we have evidence, and this is a work in progress at the moment, we're still building up N, that nanog forms regions of high density while SOX2 does not. And we also see similar behavior in the chromatin-bound species. So the take-home message here is that nanog and SOX2 do indeed employ different ensembles of strategies to maintain chromatin binding. Nanog is more lowly expressed, but it employs clustering. We think it's employing these clustering behaviors and longer residence times to overcome that concentration deficit, where SOX2 is kind of doing a brute force method of just, let's put in a whole bunch of protein, and just by probability, it's going to occupy the chromatin. So where are we heading with this? Gökçe Agsu, an excellent postdoc in Stringen's lab, is now going in to perturb the system and has mutated these tryptophans into alanines to eliminate that behavior, to see if we can ablate that clustering behavior. And then lead-ons from that is, how does this actually impact the transcriptional state and therefore sulfate. So a big thank you to everyone involved, in particular Srinjan Basu and Gökçe Agsu here and everyone else, to the BSDB poster prize, which afforded me this lovely banjo. And if I can just do a shameless self-plug here, I have just started my lab in Sheffield in the School of Biosciences, where I'm investigating early human development. So if you are interested in stem cells and developmental biology, super resolution microscopy, mathematical modeling, machine learning, blastoids, anything fun like that, we will be advertising an EPSRC Landscape Award shortly. So please contact me and thank you for your time.
Thank you so much, Stan. And absolutely, yes, come to Sheffield. It's a brilliant place.
Lovely.
So we have one question already in the chat box for Stan. Do NaNOG and SOX2 dimerize or interact with any cofactors? And if so, how might this affect their binding kinetics?
Yes, they absolutely do. So there's all sorts of pull-down studies, chip studies. If you look throughout the genome, their binding sites are near each other. In pluripotency in general, there's a whole ensemble of interconnected transcription factors that all bind near each other, with each other's, promoter, like the regulatory elements for each other. So it's quite a complex system in that regard. And this is kind of an entrance to starting to investigate those kinds of questions.
Stan, we've got 3 more questions. I'll try and get through at least a couple of them. If NaNOG needs to bind DNA in clusters, but SOX2 doesn't, does this suggest a more switch-like behavior as cells respond to increasing amounts of NaNOG, but a more graded response to increasing amounts of SOX2?
So that's a very interesting question that I've addressed through modeling with one of my old students, kind of asking about this slow breakdown versus rapid decondensation. I think I'm hoping that this W10A construct, where we're ablating that ability to form the condensate in the first, let me temper my language. I don't want to call it a condensate at the moment. There's these clusters. If we can ablate that behavior to see if we do get that switch-like behavior. But in my head, it makes sense to be able to have that rapid turnover, especially considering that NaNOG is just there for naive pluripotency for a brief period of time, and then it needs to shut down quickly and efficiently upon implantation.